Hi folks, Dr. Marbalia. Welcome to Cancer Healing Vlogs. Let's talk about cancer genomics. Now, I'm nowhere an expert in this subject. In fact, my understanding is pretty limited, but I'd like to obviously down the line get a bit more understanding because obviously genetics and all those DNA aspects are going to be the future of where our science, medical science, and treatment and health is going to go. So, in the long run, definitely one of my goals to learn for now what my with my basic knowledge and understanding I can share with you, and of course it's very basic stuff, but firstly, in cancer genomics you are talking about, you understand a genome, which is basically the entire length of the gene. If you're looking at it, if you study the normal genome, which has been done already, then you compare that with a cancer cell and its gene, and then you see, is there any difference between the two? Now, the way you can do that testing is there are these very complicated tests, which one of them is like the sequencing. Earlier there was Sanger sequencing, then we can next generation sequencing. These tests can detect, like, go through the entire gene and say, okay, this is slightly different, this is slightly different there, and it gives you basically an entire. It's a very complicated way of testing, but in simple terms, it helps you detect what is the gene of that individual cell that we are looking at. Then there is the PCR analysis, polymerase chain reaction, that whole test for that. Then there are multiple ways in which it can be looked at. One of them is also like there are panels like microarray. That's a complication which I am still not aware of exactly. Then there is fish analysis, that you're taking specific probes of a part of the gene and you're analyzing that. So different methods have been done, and there are more methods out there which are being done to. Now, what are you achieving out of this through getting an idea of, okay, this is the gene for a cancer cell? Is that it's helping in treatment, because if they are able to detect the cancer cells, then the medications and the drugs that are coming down the line are targeting those cells which have those genes and the mutations for this cancer, which are different from the normal. So if you can differentiate the normal cell from the normal cell and then get the treatment or the chemotherapy or whatever form of therapy to that required cell and not to the other cells, that's our current goal in cancer therapy, right? Also in detection. So I spoke about the therapy part of it, but then the diagnosis or timely diagnosis is also crucial, right? So if at the genomic level, there we can detect, okay, this is a cancer cell versus this is a in normal cell, even though the like the physical change has not happened, but the genetic change at the nuclear and much even more microscopic level is happening, and if that can be detected. So these are the methods that are helping. Of course, they're more expensive at the moment, and there needs to be done a lot of work and research on this, where it can be done at perhaps somewhere down the line at a much cheaper and much affordable thing for the general masses. But that's just a bit of insight which I have on the subject of cancer genomics. Of course, uh, you know forgiveness for any lack of correct information, if at all, anywhere, but do verify what I'm saying, and more importantly, if this is topic, a subject that interests you, then definitely let's go deep into this, because that's where the frontier is, the next scope is. All right, stay aware, liberate self, like and subscribe. Cheers.